Hello everyone and welcome back to Biolympian. This is my second video and today's video is going to be on the topic deoxyribonucleic acid, also known commonly as DNA. Now in the previous video I had promised some good news to come out of my NEET results, and that news has arrived. I have secured an All India rank of 541 in the NEET UG 2022 examination, and hopefully I have booked myself a seat in one of the in a reputed medical institution of our country. Okay, so I am doing this video on DNA today because my next video it's going to be on genetics, right? And before I get into genetics, it's important that you guys have some understanding of DNA, because DNA is inextricably linked to genetics. It's basically the means by which genetics progresses. Okay, now if you've watched my previous video and if you've watched some videos on my other channel Fizzy, Phizzy, Fizzy, you know that I have an obsession with the basics, right? Basic concepts of everything. So even in this video, I would like to begin by delving into the basics of it. And by the basics, I would like to explore a few biomolecules, right? A few important biomolecules, right? Okay. Now, what do you understand by biomolecules? Another long word. It's simple. It's bio as in biological, and molecule as in uh, an aggregate of elements, right? A compound, a chemical compound, right? A few biomolecules. And the first biomolecule I would like to talk about is sugar. Sugar. And in particular, about a specific type of sugar. But first, let's talk about sugars first. What are sugars? So, sugars, if you have a bit of concepts in uh, biochemistry, you might know that they are polyhydroxy aldoses as in having aldehydic groups, C double bond O H, or ketoses, right? Ketoses. And ketoses is just one, maybe R group attached to the C, one double bond, and another R dash group attached to the C. Okay, so ketoses as in derivatives of ketones. And polyhydroxy is also another long word. Poly, as you probably know, it means many, M-A-N-Y, many, and hydroxy stands for OH group, OH or alcoholic groups. Okay, so that, I think it breaks it up nicely enough for you now. Polyhydroxyaldoses or ketoses, that is, compounds that contain a number of OH groups, a number of alcoholic groups, as well as one C double bond OH group or one C double bond O group. Now, you've probably heard about sugars like glucose or fructose. If you want to see the structure of glucose, it's kind of like this, right? See, there's a C double bond OH group, and this is a COH group, another COH H, that's three C's, COH H, COH H, and another, this is 5 done, right? CH2OH. You see there are 1, 2, 3, 4, 5 OH groups, one C double bond OH group, and 6 carbon atoms. So this is polyhydroxy part. You understand it's because of so many OH groups being present, and you understand that the aldose part is because of this aldehydic group, this big aldehydic group right there. So this is an example of one such sugar, which is glucose. All right. So today we are not going to be talking about glucose, because glucose isn't what's of real importance to our topic today, our topic of discussion today. We're going to be talking about a particular type of sugar known as ribose, and its derivative. Actually, its derivative is more important, which is that or sign is wrong there, and deoxyribose. Ribose and deoxyribose. Deoxyribose is the more important of the two for our discussion today. Okay, so Probably, after seeing this diagram, you'll be a little confused, but follow me a bit here. I'm drawing a structure, and if you know bond line formulas, you'll understand why I'm not writing in the carbon atoms. So how many carbon atoms can you see here? 4, right? Each corner of this pentagonal ring structure represents a carbon atom with one oxygen atom in between. So that is not a carbon atom, it's been replaced by an oxygen atom. So there's 1 here, 2 here, 3 here, 4 here. So 4 carbon atoms in the ring. There's 1 here, HOH2C, and there's a hydrogen here. There's 1 more hydrogen here, OH. That's a polyhydroxy part coming in with all the OHs, alcoholic groups. 1 more OH, and there's an OH on the top, 1 H on the bottom. And we call this is a sugar, but this is a different type of way of representing a sugar known as a Haworth structure, right? Haworth structure. This is a Haworth structure because it's not the open chain. You cannot see the aldehydic or ketosic group in this sugar anymore, because it's formed a ring. It's formed a ring known as a hemiacetal or a hemiketal, depending on whether it's an aldose or a ketose, and that's why you don't see the double bonded carbonyl group anymore in this structure, right? This is known as a Haworth structure, and this particular sugar, we call it ribose. Ribose. This particular sugar is known as ribose, right? Ribose. Now, if you know a little bit of organic chemistry, you can also probably observe that this is beta ribose, but you need not go into that much detail. Okay, if you don't know what beta ribose is, it's not a problem, it's fine. You don't need to worry about it. All right. Okay, now I'm going to draw the same structure, but then I'm going to do something that's probably a little bit unfair to this little fellow right here. Alright, that's probably a little bit unfair to this little fellow right here, because I'm going to take one of its prized possessions away. I'm kind of vindictive, and I'm going to take one of this guy's prized possessions away. Oh my god, how can you be so evil, right? So you see, this is also beta ribose, as I drew in the previous diagram. But what I'm going to do here is I'm going to take my eraser, my robbing instrument, and I'm going to take out this OH right there, and I'm going to replace it with an H, right? Oh my god, you've taken its hydrogen, its oxygen away, and you've left only the hydrogen in its place. Oh my god. So I have taken away the oxygen. I have deoxified it, right? I have deoxified. So what do you think this will be called? Deoxyribose. Deoxy, as in I have taken the oxygen away. I have deoxygenated it. Deoxygenated it. De means to remove something from something else. So this is the Haworth structure for deoxyribose. The Haworth structure of deoxyribose, and in today's discussion it's this deoxyribose that's going to be more important than our ribose, since we're going to be talking about deoxyribonucleic acid, which is DNA, and not ribonucleic acid, that is RNA. Okay, if we were talking about ribonucleic acid or RNA, our ribose would be more important. That's kind of that's pretty easy to understand. And now let me just make the picture of deoxyribose a bit bigger, because then I can get to labeling its nitty gritties for better understanding. You see, I put in the different elements in here, elements and groups, and almost there, one hydrogen, HOH2C. Right. So now what I'm going to do is I'm going to take this little guy as my reference, right? This is the reference. I'm going to take him as my reference, and I'm going to move clockwise. Right? I'm going to take this oxygen as my reference and move clockwise. And serially, according to which carbon atom I encounter first, I'm just going to number them, right? The carbons, like the good guys standing in my way, I'm a bad guy plowing through them, and the carbon is the good guys, and the carbons are the good guys standing in my way, and I'm taking them out one by one, and I'm numbering each KO, right? So this little guy, this carbon atom right here, will be number 1. Okay, this carbon right here will be number 2. This carbon right here will be number 3. This carbon right here will be number 4. And this carbon right here will be number 5. Alright, so I've numbered the carbon atoms in my structure of ribose, and now let's see what we can do about this. Alright, let's see what we can do about this. Now first off, you may have noticed something fishy right here, right? You see, this OH is still here. Hadn't I taken it off when I went to deoxyribose? That's right, I had. Then why did I put it back here again? Because I wanted to prove a point, right? I wanted to prove a point. Now, which carbon atom do you think this OH has been taken away, the oxygen has been taken away from, and hydrogen has remains in its place? It's the second carbon atom, right? See, if you consider oxygen as a reference point, you see it's a second carbon atom that has been robbed of its oxygen and is left poor and starving, right? So we call it this carbon, we call it the 2 prime carbon, the 2 prime, 2 with that dash is called 2 prime. And what is the name of this sugar? Deoxyribose. So that is why you may have come across this term, 2 prime deoxyribose. You may not know that this dash is called prime, may not have known earlier if you came across this term that this dash is known as prime. Know it now, it's called prime. It's 2 prime deoxyribose. It's very simple, right? If you encounter this without knowing this structure, then you're kind of lost in the water, right? What does this 2 out of nowhere, how does this 2 come in front of the name of the compound, right? So 2 prime deoxyribose is because the carbon atom at position 2 has been robbed of its oxygen, of its OH group, and a hydrogen group has been placed in its stead. Alright, so this is what I require. This is my compound of the day, this is my sugar of the day, and this is 2 prime deoxyribose. So if you know the expansion of DNA, deoxyribonucleic acid, you kind of get where that how that first deoxyribo comes, don't you? Deoxyribose contributes the deoxyribo to my deoxyribonucleic acid. So you can kind of guess how of how much importance this deoxyribose is to the entirety of the compound, that it makes up almost 65 to 70% of the name, right? Deoxyribo, and the nucleic acid is comparatively a smaller bit than the deoxyribo part, right? So if not 65 to 70%, that was an exaggeration. Let's consider it's 60%, right? So 60% of the name is contributed by 2 prime deoxyribose, right? Now let's move to my next biomolecule of the day, and that is nitrogenous base. Nitrogenous base. To be precise, nitrogenous bases in the plural, as Thomson and Thompson say, to be precise, nitrogenous bases. Okay? And the first category of nitrogenous bases that I want to talk about is purines. Wow, purines. That's kind of cool name, isn't it? That's kind of a cool name. Purines. It's it's kind of a comic character name, or maybe a superhero name, sort of. Okay. Okay. So now let's draw the rather complicated structure. Don't worry, it may appear complicated at first, but with the right understanding, nothing's complicated, right? Nothing's complicated. There's two ways to something. You either know it or you don't. If you know it, it's easy. If you don't know it, it's difficult, right? And my intention is to make you know it so that it's not difficult anymore. It's that simple. Everything in life's that simple. Okay. And It's a very benzenoid structure, right? We call this a heterocyclic ring structure because it's just an addendum, sort of. We call this a heterocyclic ring structure because you see, obviously, some of the carbon atoms have been replaced by nitrogen atoms, right? So it's a heterocyclic ring structure, and it's a benzenoid structure because it resembles the structure of benzene. This benzene looks kind of like this. If you know about it, it looks kind of like this, right? It not kind of like this, it looks exactly like this. So it's just, you see, some of the carbon atoms have been replaced by nitrogen atoms. That's all there is to it, right? So it's a benzenoid structure and it's a heterocyclic structure because in the cycle some of the carbon atoms have been replaced by nitrogen atoms, and this is what we call a purine. And now let the numbering commence, right? Let the numbering commence. I am very fond of numbers, and in this case we consider this nitrogen atom to be the reference and we number it as 1, okay? And then we proceed in the counterclockwise direction. The numbering in this case is not very important, but just know it. Since I'm telling you about it, you there's no harm in knowing it, right? And just, if you just follow my stylus, if you follow what I'm writing, you can see I'm putting in the numbers, I'm plugging in the numbers as it should be numbered. So it's a 9-membered ring, right? If you can see, it's pretty evident from here. It's a 9-membered ring. So nitrogen first, then anti-clockwise, counterclockwise till number 6, and on the other ring it actually proceeds clockwise. So 5, 7, 8, 9, right? 7, 8, 9. Since the 6 is this side, so from 5 to 7, right? 7, 8, 9. It's like this, and this is the basic structure of a purine. This is the basic structure of a purine, right? And now we are going to consider some of the important purines that are going to be useful to us for learning about DNA. There are quite a lot of purines, there are quite a large number of purines, but all of them are not important to us. So we'll only consider the important ones today, okay? Now the first one, it very closely resembles uh, the general purine structure. That's obvious because it is a purine, so it must resemble the purine structure. So let me draw the general purine structure first. That's it, right? That's it. Now put in the double bonds. And now at this position there is one amino group, NH2. One hydrogen is replaced by an amino group. That's the only difference between uh, between the general structure of the purine and this particular purine. And if you notice the numbering here, this position is 6, right? So what will it be called? It will be called 6 amino purine. That's a logical way of thinking about it, isn't it? 6 amino purine, and this has a very special name. We call it adenine. Adenine, Adenine, or some of you may choose to call it adenine. That's also correct, but I think the American version is adenine and the British version is adenine. And since I'm an Indian, we follow the British version, and that's why I'm calling it adenine, right? Okay, now let's consider another purine. This one also has the basic skeletal structure. This one also has the basic skeletal structure: nitrogen, 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 hydrogen. But there's a small difference in that this nitrogen is also bonded to a hydrogen, right? And now, since nitrogen has a valency of 3, there cannot be any double bond here. And so there is one double bond here and there is one double bond here, but none here. And since carbon is tetravalent, it has to fulfill 4 valencies. And so it does this by double bonding with an oxygen atom here. And in addition, there is an amino group right here, right? And I missed out the double bond here, so I'm going to put that in, right? And if you see this structure as well, you can kind of gauge by the numbers, by the numbering system we followed in the skeletal structure of the purine. You can kind of gauge what its name is going to be. It's 2 amino 6 oxo purine, or you may call it 2 amino 6 keto purine, okay? And this one has a really cool name to it as well. It is called guanine or guanine, okay? guanine or guanine. Okay, so that is all about the purines part. I mean, not all about it, but all that's important considering our DNA discussion. And now we're going to proceed to the second category of nitrogenous bases, which are called pyrimidines. Okay, pyrimidines kind of resembles the name of the Great Pyramids of Giza. Pyrimidines. Pyrimidines. I wonder if uh, that was the intention behind the name. And pyrimidines, they have a very nice structure as well. And we look at the basic structure first, as we looked at in case of purines. And in this case, it's like this. There's two nitrogens, and you'll notice there's only one ring. It's not two rings attached to each other, right? There's one double bond and another double bond, and you see it's not exactly benzenoid because it does not have the third double bond that benzene has, but it resembles benzene because it's again six atoms, a six-membered ring. So we call it like we don't call it anything, but you may consider it to be kind of benzenoid. Okay, and let's number again. Let's number again. And this time we're going to be taking this nitrogen atom as the reference atom. Okay, and this is going to be number 1. This is going to be number 1. This atom is going to be number 2. This is going to be number 3, 4, 5, and 6. Right, so it's a 6-membered ring. We proceed clockwise this time, and this is nitrogen number 1, 2, 2, 3, 4, 5, 6. Okay, and it's fairly simple compared to purines. And now we're going to be considering some of the important pyrimidines considering our present discussion, which is on DNA, right? And the important pyrimidines in our case are this one. It has N H, and there's this double bond here, and it's slightly different from the general structure that we noted above, in that it also has a hydrogen attached to the other nitrogen atom, and since that completes the trivalency of the nitrogen, we don't put the double bond there anymore. And if you see here, since carbon has to satisfy its tetravalency, we put a double bonded oxygen atom there, and this carbon also has to satisfy its tetravalency, so there's another double bonded oxygen atom there, and here there is a methyl group CH3. Okay, a methyl group CH3, and we call this has a pretty cool name. Now logically speaking, what do you think its name should be? It should be 1,4-dioxo 5-methyl pyrimidine, right? That's it, just follows a logical sequence like that. But we don't call it that. That's a mouthful of a name. We call it thymine. Thymine, right? Thymine. Or we don't call it thymine, it's thymine only. We go by the British pronunciation on this one. Okay, and the other important pyrimidine for our study is this one. First, the basic structure: nitrogen, nitrogen, hydrogen, and there's a double bond here, and there's another double bond here. This one resembles a basic structure, if you see, to a greater degree than our previous one. And here there's an amino group. Hydrogen group is substituted by an amino group, and there's a double bonded oxygen here. Okay, and what do you think this will be called? Again, this is 2-oxo 4-amino purine, or actually 4-amino 2-oxo purine, since it's alphabetical. So 4-amino 2-oxo purine, right? We don't call it that as expected. We call it cytosine. Cytosine, right? Cytosine. Okay, cytosine. And all these purines and pyrimidines, they have We cannot represent the entire name when we are drawing a condensed formula, right? So we just call them, we just represent them by their first letters. Adenine is A, guanine is G, thymine as T, and cytosine as C, right? Okay, okay. Now let's move on to the third piece of the puzzle. It's not exactly a biomolecule because it's inorganic. It is the phosphate ion, right? Phosphate ion. Phosphate ion. And if you know basics of inorganic chemistry from maybe 8th standard or something, you know how it looks like. PO4 3 minus, P double bond O minus, O minus, 1 2 3 4, and here's the fifth one, O minus, right? This is the phosphate ion, PO4 3 minus, and in DNA studies we actually represent it like this: P double bond O O, and this O remains as minus, right? Because we represent this oxygen as covalent because it's covalently bonded to some other atom, as we will see in our subsequent studies, right? So this was all about the few biomolecules that we needed to talk about before we moved on to rigorous DNA studies. A few biomolecules, just recapping: sugar, which is deoxyribose, 2' deoxyribose; nitrogenous bases, purines and pyrimidines. Purines consist of adenine, guanine, thymine, and cytosine; and the phosphate ion, right? The phosphate ion, the important structure being this one. All right. Okay. Now, once we've got that out of the way, we can proceed to another topic, right? And the other topic, the new topic, it's going to be nucleic acids. And that's the second part of our name, deoxyribonucleic acids. Nucleic acid comprises the remaining 40% of the name, since 60% is already taken up by deoxyribo. 40% is nucleic acids. And nucleic acids have levels of organization, right? Everything everything is composed of smaller parts, right? Smaller parts contribute to making a bigger part, and these bigger parts contribute to making an even bigger part, and that contributes to the whole. Even we are composed of atoms, atoms to cells, atoms to organelles, organelles to cells, cells to tissues, tissues to organs, organs to organ systems, and organ systems to organisms, right? Nucleic acids, if I consider the first step in this classification, in this organization, it's the compound that is formed in this way. It's called a nucleoside. All right, it's called a nucleoside, and the way nucleoside is formed is by the addition of a nitrogenous base to a deoxyribose sugar. Deoxy, since we are talking about deoxynucleosides in DNA, it's by the addition of a nitrogenous base to a deoxyribose sugar. Now, how does that work? Suppose I have a deoxyribose right here. Let me use yellow. Suppose I have a deoxyribose right here. This is a deoxyribose, right? This is skeletal, rather skeletal. And I can do one thing, I can omit the hydrogens altogether. We usually do that in organic chemistry, we omit the hydrogens altogether. And in that case, you see, this is my deoxyribose, right? This is my deoxyribose. But now what I do is I take, let's say I have something like, let's say I have adenine, right? Nitrogen, N, H, N, I'm sorry, that's wrong. N right here, N, NH2, let me put the double bonds in. There's one double bond here, one here, one here, one here, right? This is adenine, right? Adenine. And now what I do is take this OH and I take this H. What does that make? What does one OH and one H make? It's water, right? And water, it's very it's cool with everything. It's a cool dude, right? We ask the water to leave. It's like, okay, fine, man. Okay, fine, man. I'm leaving. Okay, and now what happens? Since the water is left, we asked it to leave, and this reaction is a very cool name. It's called condensation, right? Condensation. I'm sorry. The water is left, and now we are left with this structure, right? Very quickly, see, the O has left, but the N is still there. OHH have left. Okay, and this I'll put in the relevant parts. Ignore my hiatus there. Um, this molecule, this compound formed by the combination of one nitrogenous base and one deoxyribose sugar, this is what we call a nucleoside, right? This is what we call a nucleoside. And just delving a little bit deeper into the nitty gritties here, what do you think? This is the what prime carbon is this? Since oxygen was a reference atom, this is the 1' carbon, right? So the nitrogenous base, we say it attaches to the 1' carbon of the deoxyribose sugar. Okay, okay, okay. Now we got it. And this bond, it's a it's formed by condensation, and it's known as a glycosidic bond, right? It's known as a glycosidic bond. Okay, and that's just a cool name. That's just a way for biologists to show off, right? It's not exactly that. That was just me being funny, but of course there's a reason to it. And but it's come on, it's a long name. It is a long name. Glyco comes from glucose, comes from sugar, and sidic is just uh, glycosidic. Therefore, stands for bond formed by a sugar. So glycosidic is a biologist way of saying it's a bond formed by a sugar, right? Okay. Now we move on to the next level of organization, which is nucleotide. Nucleotide, right? Side and tide, not to be confused, as Wikipedia says on top of its articles, not to be confused with something else. Nucleoside, not to be confused with nucleotide. All right, nucleoside and nucleotide are not to be confused. Now, nucleotide is a nucleoside which consists of, which in turn has an added phosphate ion at the 3' position, right? 3' position. Now, what do you think? This is 1', this is 2', so this has got to be 3', right? This is a 3' position. It's pretty simple when you look at it that way. So, I won't, actually, I should do the intervening steps. So, let me first take a nucleoside. Let's first, and I'm not making the base all over again. Let's just say this has a base right here, right? This has a nitrogenous base right here, and I'm drawing the HOH2C, the CH2OH, and this OH. Now the 3' OH, when there's a phosphate here, when there's a phosphate here, okay, and let's say this is phosphoric acid, right? Not phosphate. This is phosphoric acid. We're making it a little bit cooler now. Let's say this is phosphoric acid, and let's say that it's partially ionized. It's getting complicated, I know, but let's say it's partially ionized, and this is one O minus, this is another O minus, right? And what happens now is that this hydrogen sees this OH, right? And hydrogen and OH have to make water. It's kind of a compulsion for them, right? They have to make water, and the water is the cool dude. Water goes out, and you know what happens next. You know what happens next. Water goes out. And now we are left with a structure that's kind of like this. This is very deep in chemistry, this part. If you're looking for, see, if you're looking for core bio, there's no such thing as core bio. It's applied physics, applied chemistry. Last lecture was applied physics. You saw potentials and charges and whatnot, and that's all physics, right? But that's also bio, and this is almost all chemistry, right? But this is also bio, and it's pretty difficult if you don't have concepts, if you don't have interdisciplinary concepts, right? You can't be good in biology without being good in physics, chemistry, and math. Okay. Okay, so now see, there's another oxygen here, right? Oxygen, and p o o minus, o minus, right? And this is what we call a nucleotide, and this bond right here, this is known as a phosphoester bond. An ester bond is one that contains double bond oxygen and single bond oxygen attached to the same central atom, and since in this case it is phosphorus, we call it a phosphoester bond. Okay, phosphoester bond. I'm sure you must have heard of esters in organic chemistry, fruity substances, fruity smelling substances like uh, acetyl, uh, uh, acetonate, maybe that is an ester, or ethyl ethanoate, that is an ester. So in the same way, this is a phosphoester, and we call this structure as a nucleotide. Okay, so we've got nucleosides and nucleotides out of the way. Let's talk about how we name them. Okay, and this is also one more thing. This is deoxynucleotide, since it's deoxyribose. That should be pretty clear to you by now. And let me make a table for nucleosides and nucleotides and what we call them. Okay, now on one side it's going to be the nitrogenous base, and then the other two, it's going to be what we call the nucleoside and what we call the nucleotide. Okay, so first let's see the N base. Okay, and in the first case, let's consider the N base is adenine. Adenine, right? Adenine. Now let's consider the nucleoside, the deoxynucleoside to be more precise. And when the nitrogenous base is adenine, the deoxynucleoside is called deoxyadenosine. Adenosine. Deoxyadenosine. This is the nucleoside, right? And now let's move on to nucleotide. And this is called deoxyadenylic acid. Okay, deoxyadenylic acid. Next, let's move on to guanine. The nucleoside is called deoxyguanidine, right? Deoxyguanidine, and the nucleotide is called deoxyguanylic acid. Very simple. Now the third piece of the puzzle, thymine. Deoxythymidine, right? Deoxythymidine, and nucleotide deoxythymidilic acid. Deoxythymidilic acid. And now, oh, that doesn't capture, so I need to draw the lines. The last bit of the puzzle, and this time it's cytosine, so it's deoxycytidine, deoxycytidine, and we call the nucleotide deoxycytidilic acid. It kind of follows a logical sequence, doesn't it? These are very long names, admittedly, but it follows a logical sequence. If you see the nucleosides, they have ene at the end and deoxy in front, and just the names of the bases in some modified, in a little bit modified manner in between. And nucleotides, they have ilic acid at the back and deoxy in front, and the names of the nitrogenous bases in some modified form in the middle, right? Okay. Now, I would suggest you look at this table once, because you may forget these names. It can get confusing at times, but with practice, it comes quite naturally. Okay? Okay. Now, I have got that out of the way. Now, let's see how nucleic acids are actually composed, right? So, let's say I have one nucleotide here, one nucleotide, sorry, let's say I have one nucleotide here, one nucleotide here, and this is base and CH2OH, and this is OP double bond O minus OH. You'll see why I left that OH right there, okay? And let's say I have another nucleotide right here, OH H2C. You'll see why I drew it like that as well. This is the base, and there's another I'm sorry, there's another phosphate ion on the other side, and I can just depict it in this way. No need for the hydrogen anymore. Now, if you see, what is interesting about this is that these are 3' nucleotides, right? Because at the 3' position there's a phosphoester bond. But now you see there's another OH here, and it's peaking at this hydrogen atom right here, right? And it says I want to be water. I want to meet with you. I want to fuse with you, and I want to be water, right? And water, once water is formed, he's a cool dude. He doesn't stick around. He just takes off. And once water takes off, what do we get finally? We get a really nice structure, and we get a really nice structure which is kind of like this: OP double bond O minus O CH2 O O P O minus, and there's base, and there's HOH2C. Wow, this is a really nice structure. It's complicated looking, but it's really simple if you understand it, right? And this, if you see this bond, there's two phosphoester bonds, right? One on this side, one on the other side. So what is two? Di, right? Di stands for two. Di stands for two. So we call this, what do we call this? A phosphodiester bond. And this, my dear friends, is what we call a nucleic acid. Nucleic acid. And this is brilliant, bright, this is effulgent, coruscant. My, those are long words, right? This is absolutely effulgent, absolutely glowing, absolutely coruscant, and we call it the nucleic acid. This is very important, right? This is how a nucleic acid is formed. Phew, we have been drudging along for so long, and we've just got to know how a nucleic acid looks like. We might have been drudging along, but it was fun, right? Since we got to know the nitty gritties of how everything works, the very basics, the absolute from the absolute grassroots level, right? And now we know how nucleic acid looks like, and when we see this structure, we don't need to be scared anymore, right? If you had seen this structure at a glance, maybe in your bio textbook, in your school textbooks, you'd have been, oh my God, there's no way I'm remembering this, right? There's no way I'm remembering this. There's no way I'm understanding any of this. I probably, if I can do it, I'll probably read it one night before the exam and just vomit it in my examination paper, and after that I'll forget everything about it. I probably won't be able to memorize the structure anyway, so let's just forget about it, right? But now you can understand, right? You have this deoxyribose sugar nicely bonded to a base. You have phosphate ion that's extra greedy and catches on to two of such structures, these nucleoside structures, right? And this just goes on, right? This just goes on till as many nucleotides can be incorporated according to the organism's DNA. This just goes on, right? And then I can I'm sorry, I did something wrong. Then I can show it in this way as well. I can show it in this way as well. I can continue the bond of the O, and I draw this symbol right here, which symbolizes that it's going on. It's going on for a large number of units, right? And this is what we call a nucleic acid. Okay, one more thing. This is 3 prime and this is 5 prime, right? And this is 3 prime, right? Now let's for a second consider that this is the last nucleotide of my nucleic acid, and this is the first nucleotide of my nucleic acid, or you can consider it vice versa. So what do you think? In this case, the OH will be free in the 5 prime end, right? It will be OH in the 5 prime end because there's no other nucleotide to form an ester bond with here, right? So in this case, the OH will be free at the 5 prime end, and we call this the 5 prime end of DNA. The 5 prime end of DNA, because the 5 prime OH is free. And in this case, let's say this OH is free and this phosphate ion isn't there, right? This OH is free and this phosphate ion isn't there. And in this case, we call it, since this is, I'm sorry, this 3 prime end is free and this phosphate ion isn't there. And since the 3 prime end has an OH and not an OPO minus double bond O minus 1 whatnot, since it does not have a O linked to a phosphate ion, we call this the 3 prime end of DNA. 3 prime end. This is very important because it gives a sense of directionality, right? We say DNA, this you'll see in a few minutes that DNA has two anti-parallel strands running in the 5 prime to 3 prime direction, or 5 prime to 3 prime polarity, and the other running in the 3 prime to 5 prime polarity, right? So that is how we classify the polarity or the directionality of the DNA into 5 prime to 3 prime and 3 prime to 5 prime. In this case, we can say that it's 5 prime to 3 prime, even though it's kind of illogical because the other anti-parallel strand isn't there. But let's say it runs from 5 prime to 3 prime, okay? And this is our nucleic acid, and I haven't I am not calling it deoxyribonucleic acid yet because I haven't drawn another crucial part of its contributing structure, right? Okay, okay. Now, now let's move on to another important part known as, I'll just 15 should be nice, 14, known as Chargaff's rules. Chargaff is the name of the scientist. You may call him Chargaff as well. Some call him Chargaff as well, or Chargaff. Chargaff's rules. And he was a Norwegian scientist who gave a set of rules that DNA abides by. DNA is a very obedient sort of child. And the first rule is that A equals T and G equals C. That's kind of cryptic, right? You don't understand. What does that mean? Well, it's very simple. A equals T means that the amount of adenine in a particular in a particular quantity of DNA always equals the amount of thymine in the same quantity of DNA. If you don't get it by now, it basically means that whatever adenine, let's say you have, let's say this is a ribose, this is a ribose, this is a ribose, right? And let's say this is bonded to adenine, this is bonded to adenine, this is bonded to adenine. This is actually one of the ways in which the structure was reduced. So you'll see in a few minutes that adenine binds to thymine, right, by hydrogen bonds. So the amount of adenine and the amount of thymine are always equal. The amount of adenine and the amount of thymine is always equal in a given quantity of DNA. Okay, in a given quantity of DNA, the amount of adenine and the amount of thymine is always equal, and the same goes for guanine and cytosine. And what this basically implies is that adenine binds to thymine by means of the means are hydrogen bonds, which we'll learn about later. But let's just say adenine binds to thymine and guanine binds to cytosine, which is why their amounts always have to be equal. This is one of the ways in which the double helical structure of DNA was deduced, right? This is one of the ways in which the double helical structure of DNA was deduced, since it has to be if even if the helical part is not very evident here, you see it kind of follows from a logical sequence that it has to be double, right? That it has to have two strands, since adenine and thymine are always equal as a rule, and guanine and cytosine are always equal as a rule. So adenine must bind to thymine and guanine must bind to cytosine, so that they can become equal to each other, right? Since they are binding to each other, they are always equal in number to each other, right? So this is the first rule of Shagaf. Okay, and the next rule says A plus G equal to T plus C. This is also very cryptic. Now A plus G, adenine and guanine, what are they? Purines, right? Purines. Purines, okay? And T plus C, pyrimidines. So this rule states that purines always equal to pyrimidines. Purines are always equal in number to pyrimidines in a given quantity or a given amount of DNA, right? This is also a very fundamental law, and it also helped in deducing the double stranded nature of DNA, because it signifies that purines bind to pyrimidines, right? The purines bind to pyrimidines, and as we saw in the last point, adenine binds to thymine and guanine binds to cytosine. Okay, and now we come to the third law of Shagaf, which states that the ratio of A plus T by G plus C is fixed and is a characteristic for a given species. All right, fixed for a given species. So whatever individual of a particular species, when we inspect its DNA, if we take the time to inspect its DNA, we'll find that the ratio of A plus T means the ratio of adenine and thymine to the ratio of the amount of adenine and thymine to the amount of guanine and cytosine is always fixed, and this way we can actually use this as a criterion to differentiate organisms of different species from one another. You see, with improving techniques of molecular homology, the lines of difference between different species have become pretty slim, right? And so we have to find something that's um, that's very rigid and unchanging as a criterion for mapping the differences between species, and this is one such criterion that we can use to differentiate between species depending upon how what the composition of their DNA is. Okay, so this is the third law of Shagaf, and now I feel it's of utmost importance to speak of the implications. All right, implications. Okay, implications. Now there are only two major implications. I say only, but both the implications are by themselves quite path breaking. So the first implication we're going to consider is that adenine binds to thymine via two hydrogen bonds, two H bonds. Okay. Now the Shagaf's law actually does not speak anything about the number of hydrogen bonds between adenine and thymine or between guanine and cytosine, but this is an experimental, not exactly experimental, but it has been deduced later. Okay, but the major implication is that adenine binds to thymine, right? And what are hydrogen bonds? What do you know about hydrogen bonds? See, hydrogen, when it binds to some really electronegative elements, say oxygen or nitrogen or fluorine or any other element of the sort, actually not any other element of the sort, basically these three elements only, really electronegative, you see it gains a little bit of positivity. The sigma bond containing the bond pair of electrons, it shifts more towards the electronegative non-metal atom, right? It shifts more towards the electronegative non-metal atom, and so hydrogen becomes very slightly positively charged. Now, hydrogen, being slightly positive, it needs that sort of negativity in its life, right? If you are too positive, you need sort of little bit of negativity to balance things out, right? Because as Thanos said, there should be perfect balance in everything in the universe, right? So when you are too positive, or if you're even slightly positive like hydrogen is, you need a little bit of negativity to balance things out. And where does it gain that negativity from? It gains that negativity from some other very electronegative elements such as oxygen, nitrogen, or fluorine, which possesses lone pairs, or a single lone pair or lone pairs of electrons, and it gains, like, it is sort of attracted towards those electrons since it is very slightly positive, and the lone pairs of electrons being present grant the electronegative atom a slightly negative nature. So since opposites attract, the slight positive and slight negative, they attract each other and form a weak bond that is weaker than a covalent bond but still strong enough to find use in our criteria of classification, right? So hydrogen bonds, it is that sort of bond which is formed when hydrogen, being positive, wants to relieve its positivity with a little bit of negativity. All right, and G being guanine, binds to cytosine. Now I don't know why I'm writing A and G for adenine and guanine, but the whole names with thymine and cytosine. But since I've already done it, I'll continue doing that with three hydrogen bonds. Now hang on a minute, isn't that some sort of discrimination or something, or some sort of disparity? We live in an egalitarian society, right? We cannot afford that sort of disparity. Now where does that arise from? No, it's not. It's not really disparity. It's nothing like that. You'll see where it arises from. You'll see where it arises from once I draw the structures, right? So this is the structure of adenine. Wait, here it is. I'm sorry, I went wrong. This is nitrogen here, N H N N. All right, NH2. Okay, and this is how we depict thymine, right? One nitrogen, another nitrogen, one hydrogen, and one methyl group right here, tucked away in the corner, double bonded O, one hydrogen right here, double bonded O. Okay, now if you observe this little guy right here, this hydrogen atom right here, it's slightly electropositive, right? Because it's got this nitrogen atom, very rowdy nitrogen atom taking away its electrons, and this poor guy, he's slightly electropositive, needs a little bit of negativity in its life, right? Needs a little bit of negativity just to balance things out, and so what it seeks is a little bit of electronegativity, and who can provide it that electronegativity? This oxygen that has two lone pairs of electrons plus is highly electronegative. This oxygen can provide it that negativity, and so it just binds weakly to the oxygen, and this is what we call a hydrogen bond. That is what we call a hydrogen bond. And you see another hydrogen right here. It's also slightly electropositive, needs some negativity in its life, and it's bound to this really rowdy nitrogen atom right here that's taking away its electrons. And since it needs that slight electronegativity, that negativity in its life, it sees this other nitrogen atom right here that's also electronegative, that also possesses one lone pair of electrons, and it just binds to it by means of another hydrogen bond, right? So two hydrogen bonds between adenine and thymine. Okay, two hydrogen bonds between adenine and thymine. Now let's see what's the case with guanine and cytosine. Nitrogen, nitrogen, hydrogen, double bond, nitrogen, nitrogen, double bond, double bond. There's an oxygen here double bonded, and there's a hydrogen bound to the nitrogen, and this is NHH. This is guanine, right? And now I'm drawing cytosine. This is nitrogen, not nitrogen, it's nitrogen, and another nitrogen, and one double bond here, one double bond here, nitrogen, hydrogen, hydrogen, and oxygen, right? Now let's just quickly pick out the hydrogens who are in need of negativity in their lives, and that is this hydrogen right here, this hydrogen right here, and this hydrogen right here. And who can provide them the negativity? This oxygen right here, this nitrogen right here, this oxygen right here. And now we quickly join these two centers of positivity and negativity together, and we constitute hydrogen bonds, three hydrogen bonds. So you see, this is why guanine bonds to cytosine via three hydrogen bonds and adenine bonds to thymine via two hydrogen bonds. Will you ever forget it now? Of course not, right? It's very simple, and it's very intuitive once you consider the structures. That's why I always say that you need to go into the grassroots of any topic you are learning, right? So adenine binds to thymine via two hydrogen bonds, guanine binds to cytosine via three hydrogen bonds, and this is how it works. Now the second implication is that DNA is double stranded. Why is that? Because adenine is binding to because the amount of purines and the amount of pyrimidines is equal, right? That can't be just a coincidence. Like you observe whatever DNA you observe, whatever DNA you pick out from any individual organism in any corner of the world, you'll see that the amount of purines in its DNA is equal to the amount of pyrimidines in its DNA. That can be a coincidence. That can't be a coincidence, right? So how do we explain it? We explain it by considering that there's two strands, and the purine of one strand binds to the pyrimidine of the other strand. So in that case, purine will equal pyrimidine. Let's say there's a purine. Let's say there's a purine here, there's a purine here, there's a purine here, there's a purine here, and there's a pyrimidine here, pyrimidine here, pyrimidine here, pyrimidine here. So four purines, four pyrimidines. So that's how purines equal pyrimidines, and that is how we know that DNA is double stranded, right? So these are the two major inferences which can be drawn from Chargaff's rules, right? DNA is double stranded, and that adenine binds to thymine and guanine binds to cytosine. Okay, so these are the two major inferences. Now we finally go to the juicy bit right here. You've seen probably DNA like this, right? Like this. Oh, there's that spiral staircase sort of thing. There's that spiral staircase. The double helix, the right-handed double helix. For some of you who may have read a few books, right-handed double helix, right? And this is how it looks like a spiral staircase, right? Like a spiral staircase. We are doing the real science now, right? We are doing real science now. And you see there's like the rungs of a ladder between them. Okay. And what are these? These are the nitrogenous bases. What are the blue and red strands? What do they represent? They represent the backbone of the DNA. And you know what the backbone is formed by? If you look at the structure of nucleic acid, which I drew earlier in detail, you will see that it's composed of deoxyribose and phosphate ions, right? It's composed of deoxyribose and phosphate ions. And the yellow lines, they signify the nitrogenous bases bound to each other via hydrogen bonds, and adenine is bound to thymine by two hydrogen bonds, and guanine is bound to cytosine by three hydrogen bonds, right? And that is basically what this rather oversimplified schematic diagram of DNA represents, right? And let's just quickly number. I told you about 5 prime and 3 prime, right? So let's say this is the 5 prime end and this is the 3 prime end, and since these two strands, as I said, are of anti-parallel polarity, anti-parallel directionality, so the 3 prime end will be bound to the 5 prime end of the opposite strand, and the 5 prime end of one strand will be bound to the 3 prime end of the opposite strand, right? So this strand is proceeding in 5 prime to 3 prime direction, this strand is proceeding in 3 prime to 5 prime direction. It's that simple. And a few parameters. The pitch of bDNA, this sort of DNA is known as bDNA. The pitch of bDNA, bDNA is the most common and most stable form of DNA, which is most commonly found in all organisms. The pitch of this bDNA is 34 angstrom, right? 34 angstrom or 3.4 nanometers, right? 34 angstrom or 3.4 nanometers. It's a simple conversion really. And what does pitch mean? Pitch means one complete turn of the helix. Now, if you have a screw, you see the groove in the screw, right? Let's say this is a screw, so the screw head will have a groove, right? Now let's say the groove was at this position when you start turning it around, right? You start turning it around, there will come a time when, let's say, I have put a single crystal of some dye in this side. There will come a time when this entire thing will turn by 180 degrees and the crystal of the die will return to its initial position, right? But the screw has progressed by then. The threads of the screw have progressed by then, and so we say that we have completed one full turn of the screw, right? So one full turn of the screw, the distance by which the screw progresses forward is known as the pitch of the screw, right? In a similar way, since screw is also a helix, it's a single helix, and DNA is a double helix. We consider the DNA to be a screw, and then the distance between two consecutive, let's say, actually, you have to use a screw analogy to understand this. So, let's say if you're holding the DNA and you're turning it, and you turn it 360 degrees in your hand. So, the distance by which the DNA will progress, or the distance between, we call these the major grooves. I hadn't come to that yet, that's why I didn't want to use that word. So, the distance between two major grooves is known as the pitch of DNA, right? And the pitch of bDNA is 34 angstrom. And since I've told you about major grooves already, let's continue with that. This is known as the major groove, major groove, and this is what is known as the minor groove, right? Major grooves and minor grooves. Once I tell you the names against the flow of how I was meaning to tell you about it, it is sort of lackluster, but that's all right. You win some, you lose some, right? So this is the major groove and this is the minor groove. As you can see from the indentations, it's pretty evident why we call it the major groove and the minor groove, right? And this distance, this is known as pitch, which is 34 angstrom, right? 34 angstrom. And let's have another point here. In one pitch of bDNA, in one pitch of bDNA, there are 10 nucleotides, right? There are 10 nucleotides. This is also a fact. And so I think you can pretty much gauge the distance between two nucleotides. What will be the answer? Distance between two nucleotides. Distance between two nucleotides. It will just be equal to 34 by 10, right? In terms of angstroms, 34 by 10, since there are 10 nucleotides in one pitch of bDNA, and one pitch equals 34 angstrom. So that just equals 3.4 angstrom, right? These are some important facts because you can use it to calculate the length of DNA, right? And calculate the if you already know the length of DNA, you can use it to calculate the number of base pairs you have, the number of nucleotides you have, right? So that's why these are important parameters. And the distance between the two strands of DNA, this is not as important as the pitch. Distance between two strands of DNA, this is 18 angstrom, 18 angstrom. In some books you'll also see 20 angstroms, but 18 is a bit more accurate, is what I perceive. 18 is a bit more accurate than 20. So 18 angstroms is the distance between the two anti-parallel strands of bDNA, right? The two anti-parallel strands of bDNA. Okay. And this double helical structure of DNA, do you know the names of the scientists who actually gave this and who actually proposed this structure and received the Nobel Prize for it? Actually, it's such a path-breaking discovery. It's such a groundbreaking discovery. It actually uh, they were actually awarded the Nobel Prize for it, right? And The names of the scientists who proposed this model are James Dewey Watson and Francis Crick. Okay, they were given the Nobel Prize along with one other guy named Maurice Wilkins, right? Maurice Wilkins and one of the stalwarts, one of the major role models for women in science is Rosalind Franklin. Rosalind Franklin and her student Maurice Wilkins was her student. They were the ones who actually observed. They actually took photographs, X-ray by means of X-ray crystallography. They took photographs of DNA, and that's how like Watson and Crick actually inferred that this double helical structure is what DNA follows, right? So Rosalind Franklin and Maurice Wilkins also did path-breaking work, and Maurice Wilkins also got the Nobel Prize along with Watson and Crick, but unfortunately Rosalind Franklin could not because she had died 4 years back. She had passed away 4 years back, and the Nobel Prize is not awarded posthumously. So it was kind of unfortunate for her because she is like one of the primary innovators in this field, right? One of the primary innovators, and all the subsequent work of Watson and Crick and even Maurice Wilkins, it stems from original photographs, from original X-ray crystallographic deductions of the structure of DNA that she took experimentally, right? And I think that should be enough about DNA. And we will speak about DNA replication also in detail when we start molecular genetics. This is just an introduction to what DNA basically is. And now I think we know enough to start with genetics, and we will do so in the next class, in the next video, in the next lecture. And till then, see you, stay safe, and all the best. Okay? Thank you.